Great. So my name's Kira St. Pierre. And for those of you that don't know me, I'm a postdoctoral fellow at the Institute for Oceans and Fisheries. And so today I'm thrilled to introduce Dr. Amina Schartup as our presenter. When I saw kind of the call for speakers, one of the kind of major themes for the UN Ocean Decade was this clean ocean where sources of pollution are identified and removed. And so Dr. Schartup fits amazingly in that theme. And that was one of the reasons that I suggested her for the seminar series this year. Dr. Schartup is an assistant professor at the Scripps Institute of Oceanography in California. She holds a PhD in oceanography from the University of Connecticut, a master's in science in geochemistry from the Institut de Physique du Globe de Paris, and a Bachelor of Science in Chemistry from Paris Descartes University. So she's a true interdisciplinarian. Before coming to Scripps, Dr. Schartup was a research associate at the Harvard School of Public Health and the Harvard School of Engineering and Applied Sciences. She also spent 2 years as an American Association for the Advancement of Science and Technology Policy Fellow at the National Science Foundation's Office of Polar Program Arctic Section, during which time she developed a federal guidance document on pursuing ethical research in the Arctic. Dr. Schartup's experience both in biogeochemistry and in policy has really put her at the forefront of research on neurotoxic methylmercury in the world's oceans, covering everything from kind of very small scale biogeochemical dynamics that control the speciation and bioavailability of mercury in aquatic environments to large scale ecosystem models tracing the effects of climate change in fisheries on the bioaccumulation of methylmercury through marine food webs, some of which I'm sure she'll cover here today. So thank you for accepting the invitation, Dr. Schartup, and I'll leave it with you.
Thank you, Kira, for this fantastic introduction. Very flattered and really excited to be here today. I've seen a lot of amazing work coming from your institution that overlap with some of my own interests. So it's really a great honor to be here. So before I kind of jump into the science, I wanted to introduce myself and tell you a little bit about why I'm doing what I'm doing and why I'm interested in it in the first place. So my mom is from Baku, Azerbaijan. So I was born in USSR, in the Soviet Union, a couple years ago. And my favorite breakfast was a slice of sourdough or rye bread and the famous Caspian Sea beluga caviar. And I know, but because as you can imagine, the exploitation of Caspian Sea beluga and the collapse of the Soviet Union that increased export throughout the world, it became rarer and rarer. And by the time I turned about 12, it was I think that's when I had my final slice. So after spending a few years in Baku, my mom and I moved to West Africa, where my dad is from. And actually, we moved to Mauritania more precisely. And I was told in school that Mauritania was one of the most bountiful seas in the world. They had a lot of fish, which now makes sense. It's an upwelling region. So indeed, there's a lot of productivity. And it was a fantastic place to grow up. And we actually lived in a neighborhood that was nicknamed Little Russia. And so funny story, I actually learned how to write in Russian when I moved to Mauritania, because when I left the Soviet Union, I was 6, so I couldn't really write really well, but I learned there because there's a lot of a big Russian community. And so there were many of us who were mixed race children. We spoke Russian, run around the desert and so on. And the reason why there's such a big Russian community in Mauritania was because right after independence from France, the Mauritania struck a deal with the USSR in exchange for access to fisheries. They would send their mostly sons to get educated in USSR. And so that's how come a lot of them went out there, and they occasionally brought Russian wife. And so that's why there were many of us mixed race children. So of course, it didn't take very long before we started hearing of overexploitation of the coast and small local fishermen having to go farther and farther out to be able to collect the fish that they needed for subsistence living. So it was kind of funny to think about it, both at the time and later on, how the socioeconomics and the fisheries and my own existence, right? Because if they were not sending those people to get educated in USSR, we wouldn't exist, right? So it was kind of like a funny thing to think about those environmental issues, but in the context of society and my own contribution to those issues. Anyway, long story short, I'm interested in the mercury cycle, but it's mostly driven to the fact that I'm interested in how human behavior and environment and the decision-making process, it really connects it, and really understanding the science, but not really without putting in the broader context of what it means for society and human health. Most of the work that I'll be talking today has been published, so I'm not listing here the authors, but there has been some unpublished contributions that I will mention. And so the authors here are Noelle Selin and Helene Angot, Kathleen Bowman, Theodore Winston, and MeHg. Those last two actually my students. So for those of you who don't know, I'll give you a brief introduction of the mercury cycle, and then we'll move on to talking about some of the projects that I've been working on, mostly in a chronological order. I find mercury really fascinating because it ties this, like I said, the industry, environmental change, Earth biogeochemical cycles, but also has this whole human history, community, and individual health component to it. Mercury is about 3,000 years of use. In its mineral form, in this bright red pigment that has been found in Egyptian tombs and ancient Chinese pottery. But as many elements and as many contaminants or usages in general, they've substantially increased since the Industrial Revolution. And now mercury has been widely used in industry, and you may have heard of mercury usage in gold mining. Those CFL light bulbs have mercury. A lot of the UV bulbs have mercury. So there's like a really wide breadth of industrial use. Inorganic mercury, so the form of mercury that you find in most of these products, is actually not that toxic because it has a low absorption rate in organisms. However, mercury vapors, you may have heard of the mercury use in the felting industry and what it did to neurological health of the workers, as well as organic forms of mercury, methylmercury, which is actually the one that people refer to when they speak about mercury in fish, is actually talking about that organic form of mercury. It has a high absorption efficiency in biological tissues, and it primarily impacts the central nervous system. So any higher level animal that has a central nervous system is particularly at risk. Fortunately for us, mercury is emitted into the environment through those industrial activities, with coal burning and artisanal gold mining being some of the biggest sources. And it ends up in fish, and it impacts humans through consumption of fish for the most part. However, the system is complex, and it doesn't go directly from emissions to consumption by humans. It goes through a range of processes in the atmosphere, deposits on land, it cycles in the ocean, it is transformed microbially in the environment, and then it enters the food web, where then food web processes and dynamics impacts this ultimate exposure. So as you can see, at every step of the way here, there are additional levels of complexity, because these are interaction with the environment that we all know is changing. So I'm interested in actually almost all of the aspect of the mercury cycle, and I've worked on some aspect in most of these steps. Another level of complexity in the mercury cycle is that the usages and emission and releases have changed over time. So you can see here, this is work by Hannah Horowitz, looking at global releases and emissions of mercury since 1850s. And as you can see, as we approach today, the complexity of the sources of mercury into the environment have increased. And so, of course, that type of complexity also comes with the fact that controls are becoming more difficult because there are different sources of mercury. But let's say we managed to get this under control and reduce releases of mercury into the environment. We're also running into an additional little problem, is that mercury has this grasshopper behavior where once it's emitted, deposits, it has a redox cycle that allows it to be re-emitted. So it kind of bounces around the environment and takes a little while to then be buried in the sediment. So Helen Amos did some work trying to replicate this through a model, and she replicated this effect of releasing mercury into the atmosphere and then tried to see how long it takes before it's buried and enters the geological cycle, right? And so this, at this time zero here, she emits mercury into the atmosphere, and then, as you can see, it takes 100 to 1,000 years for it to start getting buried in those sediments, which means that that mercury right there that we emitted a century or two ago is still cycling through the environment today. It also means that the mercury that we're releasing today is going to be cycling in the environment for the next 100 or so years. That means that we are going to have an overlap between those environmental and global changes that we're observing and the mercury that we are releasing today. So we estimate about 2,000 tons of mercury a year are emitted through anthropogenic activity. This mercury then has this, like I said, grasshopper effect when it bounces around before it reaches the ocean. Once it reaches the ocean, that's where it is transformed into the organic species of mercury, which is methylmercury, and then it biomagnifies in the food web. So at every step of every trophic level, the concentration increase, and they tend to reach fairly high concentration at the top of the food web, particularly in high trophic level predatory migratory fish species such as tunas. And tunas end up as a result, because people like to eat them and they have high mercury levels, being a major source of methylmercury exposure. This is an example for exposures in the United States, where tuna is pretty much almost half of all the US methylmercury intake. Shrimps come second. They're not particularly high, but people eat a lot of shrimp, so that makes up for it. So I'll walk you through a few examples of some work that we've been doing, looking at the impact of industrial development following hydroelectric reservoir creation and human exposure to mercury for the community living around the system. And another example, predicting fish mercury content as a result of environmental change. And then finally, I'll talk briefly about some of the work we're doing for the AMAP Chapter 8 report that is currently in prep. In my previous group, we used a combination of tools. So we do field work. So we often collect our own data that we can then use in model evaluation. And so it's kind of, in a way, allows us to develop models, realize where are the gaps in terms of evaluating model data, and be able to collect the data that is directly relevant for model development, rather than trying to gather from other work data. It can be somewhat mismatched in terms of timing and so on. So we use our in-house analytical capabilities to do the work with a set of really low trace level specialized instrumentation. We work on model development, as I've mentioned, both on a regional as well as a global scale. And we remain connected with the policy, as well as doing some risk assessment work that allows us to connect to what are the needs on the ground in terms of policy, as well as, I'm looking for the word. Forgot the word, sorry. it happens sometimes. So example number one, we're looking at evaluate the impact of reservoir creation on a downstream estuarine fjord. And this was really driven by the Nazi government interest in trying to understand what is the impact of a planned hydroelectric development on 3 communities around the downstream environment of Lake Melville. It was driven by their understanding that of previous cases of exposure by indigenous communities living near reservoirs due to an increase in methylmercury in Canada as well as in Brazil following the development of reservoirs. And the reason for that is because, as I said, mercury gets emitted and then deposits into the soils. Most of the time when it deposits in soils, it doesn't really do much, just sits there bound to organic matter or other minerals. And but once you flood that area, when you create a reservoir, you create the right conditions for the microbes that are living in there to transform that inorganic mercury that was just sitting there into methylmercury. And there's a long history of well-documented cases of substantial increase in methylmercury levels in those freshly built reservoirs, and the communities living around the reservoirs that rely on the fish that lives there being exposed to extremely high levels of methylmercury. So in that context, we're interested in finding out whether these impacts of a mercury production, increased methylmercury production, will be seen downstream in a downstream environment from the reservoir. So this is the system. It's Lake Melville. It's not a lake, it's an estuarine fjord. And the reservoir is planned right there in Muskrat Falls, which is about here. And the downstream environment is where we have several communities living here: Happy Valley, Goose Bay, Northwest River, and then Rigolette, with Rigolette being the community that is the most reliant on fishing and hunting around the system. And so what we were trying to do in this particular case is that we needed to link both the human component as well as the geochemical component and try to have some prediction, some ways to predict what are the trends are going to be following industrial development. So there were like 3 parts to this. One, we needed to understand the baseline geochemical conditions of methylmercury production in that system pre-reservoir creation, right? So we went out and collected atmospheric measurements, water, sediment, river inputs, and looked at how the mercury cycles in that system to understand that baseline level. Then we did incubation experiments. So you're seeing some of those incubation cores here around the area that they planned on flooding. So we took cores and we used the river water that was going to be used to flood that reservoir to estimate, to actually measure by how much methylmercury will increase as the water incubates the soil from that system. We then use this information to force a model that looks at a pulse of methylmercury following that reservoir creation through that baseline system that on which we created a model. So we combined that with baseline methylmercury exposure level in the community living around the area. So we obtain dietary information from the community. So they told us, how much fish they ate, where the fish was coming from over a certain period of time. They provided hair samples. So we knew what their baseline exposure was and have an idea of from which species were they getting most of their mercury. And I'll get to that in a second. And so the next step was, once we had all these baseline information, we developed this probabilistic simulation of what will happen if you flood the system and by how much mercury going to increase in the river downstream of the reservoir, as well as the estuarine surface. And we estimated it on average that it will be around 10 times increase in the river and about 2.5 times increase in the estuarine water. The range here is consistent with what has been observed in other systems post reservoir creation. We then propagated it to the animals and the food that people consumed from the system. So we received a dietary survey, so we know what they eat. We also obtained samples from fishermen locally from the different areas of the system, so we could measure the concentrations at baseline. We then estimated based on how much each species spends in the different parts of the system, whether it's they're spending most time in the river or in the estuary or offshore, so no increase there, but a little bit of an increase in the estuary, a huge increase in the river, right? We can then calculate how much we expect the increase to be in the different species that the communities that live around Lake Melville were consuming and exposed to. And so we had both baseline exposure as well as our post-flooded exposure, assuming that consumption of the different species that people are consuming at baseline will not change in the future. And then the other thing that Ryan actually did, so this is in a somewhat separate study, was to look at what is the contribution of traditional foods for methylmercury as well as all the other things. So we really needed to be careful and work this line where while we are investigating the impacts of the hydro development and looking at how much methylmercury exposure will come from the food, is also to really highlight that traditional foods is still a good source of the healthy stuff, and that people probably should not replace it with something else that is less healthy than continue to consume traditional foods. So between the measured hair concentration and the modeled concentration, we found a good agreement. So we obtained the model hair concentration from the diet that people provided. So they would say, okay, I ate, lake trout and salmon and so on. And then we measure the hair, and then we have a good agreement between the two. And this allows us to then use the model to predict future exposure, assuming that people continue to consume the same diet post-flooding. And what we found here is that at baseline, quite a bit of the population was already at kind of the limits that are proposed by the EPA in terms of do not exceed those, otherwise you'll start seeing health impacts, and that post-flooding, that these could substantially increase for the section of the populations that consume a lot of the traditional country foods from the Lake Melville regions. And that I had some data, but I'm not showing it here. It was mostly populations, like the older segment, the older demographic that tend to consume more of the traditional foods versus the younger people had a more varied, less traditional foods heavy diet. And so they were a little bit less at risk in terms of increased methylmercury concentrations. And so what the idea was there is that we need to push for a monitoring of methylmercury levels in the system and thinking about ways to flood and create the reservoir to limit the increase in methylmercury. Because as I've mentioned earlier, the traditional foods are also a source of all these other good nutrients. So the traditional advice of just change your diet is not something that is recommended, both for health reasons, because it provides vitamin D and so on, but also for cultural reasons, because you don't want people to change their traditional ways of living just because they decided to dump a bunch of methylmercury in it, pretty much, right? So the take-home message from this work was that we developed an analytical framework that links the geochemistry to biota, but also to human exposure, and proposed some reservoir designs that minimizes impact on communities that rely on country food, such as run-of-the-river reservoir creation, flood as little as possible, do not flood a lot of land right away, just kind of like increase it slowly so you don't have this big pulse of methylmercury that goes into people's waters. And really kind of like the philosophical point here is that because of the scale that we're looking at, energy production, regardless of the type, has an environmental and human cost. So even though hydro development is considered a green energy source, we really need to think about who is going to carry the burden of those shifts, right? So while at a global scale, yes, we want more hydro because it's better than burning a bunch of coal that releases mercury too, right? I think that it's really important to think about who are we shifting the burden of those global issues that we're dealing with. And very often the burden disproportionately impacts indigenous communities. And so these are communities that are often already at greater risks for environmental change. And so we start going into an environmental justice concern here that we really need to be aware of as we're trying to find solutions to climate change. So the second example here is looking at predicting fish mercury content following declines that we are seeing in emissions in North America. So this is an example from work from Yang Xu, where he modeled declines in elemental mercury in the North America, mostly driven by a better control on US utilities of some other contaminants. So we had a lot of co-benefits due to control of NOx's as well as sulfur emissions and switch to natural gas. And this resulted as a kind of like a benefit in reducing mercury emissions in the northeast of the United States. So the question was, we're seeing those declines over the past several decades. Are we seeing declines in fish mercury levels, right? Because if we are assuming that we control the mercury emissions, we will see a decline in fish. Then we need to know by how much and if that's the case. And one of the things that I was concerned about is there's all these other environmental changes that are happening in this region at the same time, such as seawater temperature increasing, changes in productivity, inputs of organic matter into the North Atlantic Ocean. So the question was, how does mercury levels in the fish change, knowing that we have all these other changes at the same time? So in order to address some of that, we developed a food web model that is sensitive to these environmental changes and used it to explore the impact of overfishing, increased productivity, or changes in productivity, food web structure, seawater temperature, etc., on fish mercury levels. So the model is based in the Gulf of Maine. And the reason for that was because we have a lot of data that NOAA has been collecting on fish stomach content, just changes in the food web structure. And then we had enough mercury in fish data to evaluate the model. And so our kind of like the top of our trophic level in the system was Atlantic bluefin tuna. And then we also had swordfish and a few other large predatory ones at the top. And we, as you can see here, had a pretty good amount of data. Each data point here is a bluefin tuna. And we kind of worked with trying to understand, like, what are the drivers of these changes of mercury levels in the fish? Is it concentration in seawater? Is it the organic carbon content? Is it the bioenergetic of the fish or the structure of the diet? What are the different fish are eating? And we worked through that for different species, and we did a pretty good job at modeling all the fish that we had in the food web model. And so the idea there was that now that we have a functioning food web model and we know how the fish kind of acquire the mercury by eating each other under the different conditions that we can change in the model, we can then try to see what are the possible changes over time and how do they impact the mercury level in the fish. And in a way, we were hoping that it would explain why fish species that live in the same ecosystem, so in theory, seeing the same declines or increases of mercury levels, how come they respond differently sometimes? Some of them see an increase in mercury levels versus others are seeing a decline, even though they live in the same ecosystem that is overall seeing a decline in deposition. So this is some of the things that we were running into that we're trying to explain with the model. And so an example here is cod mercury level over time. what happens if you increase the model seawater concentration, seawater temperature by 1 degree, you see that cod mercury levels increase just as the temperature of seawater changes. However, if we switch the diet from cod for what we had at base, which was the 2000 diet, to a 70s diet, you see that the 70s diet actually had a lower methylmercury in cod than the 2000 diet. However, when we did the same thing for dogfish, we saw the opposite trend where the 70s diet, from the 70s to the 2000, we see a decline in mercury level. This is fish that are living in the same ecosystem, exposed to the same waters, the same mercury levels in the water, but they're seeing that decline. And that's just driven by the fact that as the herring, as you remove certain species from the ecosystem, so in this case, it was herring, those two species that used to rely on eating herring switch to a different prey. And so depending on the prey that they pick, their mercury levels go in different directions. So we thought that was really interesting. But overall, the temperature increase on all the species that we look like tend to increase the methylmercury levels. We did something similar for Atlantic bluefin tuna, except that in this case, we had kind of like an historic, we forced the model with historic seawater temperatures as well as methylmercury concentrations, and look at how the combination of both changing seawater concentrations of mercury as well as the temperature impact the bluefin tuna methylmercury levels. And what we found was there were ups and downs driven by both the concentration of mercury as well as changes in temperature. But one thing that was interesting is that when we projected into the future and kept the methylmercury in seawater almost constant, so that dotted line here, we see an increase in methylmercury in bluefin tuna just driven by warmer temperatures. And so the warmer temperatures are impacting the bioenergetics of the fish and really the bioenergetics in the entire food web. As you can see, this fish below it see their increase in methylmercury due to increased seawater temperature, and sitting on the top, the bluefin tuna kind of integrates that whole signal from the bottom of the food web. And so what this is telling us is that we want to see more aggressive emissions control, that we can't assume that if we reduce emissions by 10%, we will see a 10% decline in methylmercury level in the fish. What we're going to see is that these other environmental changes may either enhance or offset some of those changes in methylmercury concentration. So we really need to understand those other environmental parameters in order to effectively reduce methylmercury level in seafood. So the take-home message here is, again, that despite declining mercury emissions in North America, we don't know whether we're going to see lower fish levels, because we don't really have a good grasp of how these kind of ancillary parameters are driving changes in uptake and so on of methylmercury. And the food web structure itself is important to be able to quantify that. One thing we know for sure is that global climate action is needed to address some of these even ecosystem-specific responses, because the increase in temperature in the Gulf of Maine is a global climate action issue, that the temperatures are rising, and so we need to have a control on that. The other thing is that those two are related, right? The temperature are rising because in part we are burning fossil fuel, the same fossil fuel that is emitting mercury into the environment. So if we have those under control, the mercury emissions will also decline. And this is kind of my segue into my final example is, well, what is the future of mercury in the Arctic following changes in those emissions into the environment? And so what we did here, so the leads for this chapter, Noelle Selin and myself, and then we have contributing authors. And so we used several anthropogenic emission models. In this particular case, we used these three, which was the A1B1, which is no action, assuming we're not doing anything to reduce mercury emissions. We have two control scenarios. And then we use the deposition from this emission scenarios to drive a model, relatively simple model, box model for the Arctic. It was based on Ann Sorensen's 2016 GBC paper. And to look at how those different scenarios will be then reflected in the different layers of the Arctic Ocean. And what we found was that, so this is, I'm going to walk you through these three. So this is the surface, this is kind of mid-depth, and this is the deep Arctic Ocean. And here in brown are the no actions, and this shows the relative change in mercury. So if you see it on this side, it's an increase. If you see it on this side, it's a decrease. And these action means, what if we act in 2020? Or what if we act in 2035? It was 2020 because at the time it was 2019, we were working on that. So we're already behind the times. It's just crazy. And so what this is showing is that in all cases, we will see an increase in mercury in the Arctic Ocean, whether it's in the surface of the ocean, if we don't do anything. However, if we do act, we will see an overall decline, even if our action is relatively modest. Although the sooner we act, the more of a difference we can make. And that's driven by the fact that mercury builds up, which was told you earlier, it's a legacy thing. So the stuff that we're releasing today is adding on tomorrow. So the longer we wait, the more we will build up in the system. And so there's also a lag time as we go with depth, because as you can see here, 2020, 2050, for the deep ocean, there's not much of a difference. That's because, well, it takes a while to impact the deep ocean. And of course, I was curious to see what is the influence of climate sensitive variables on mercury. And we know that in the Arctic, sea ice is changing, river input is increasing, and primary production has been increasing too. And so we wanted to see how are these other changes going to impact mercury level. We know that sea ice is kind of serving as a cap on the air-sea exchange. So maybe removing sea ice, we'll see more evasion of mercury. I don't know. So that was the idea, kind of playing around with those environmental parameters and see what we see. And what we are seeing is it's a similar plot. So this time, like you said, surface, mid, and deep ocean, but this time we're looking at the impact of sea ice, river, and net primary production, and is also all of them together. As you can see here, the scale is smaller than what we had with the emissions. So what it's really saying is that those environmental parameters have an impact on the mercury levels in the Arctic Ocean, but it's smaller than the emissions control. So we still, again, go back to, we want to control emissions of mercury into the environment. So the sea ice really has the largest impact here on the mercury level, and the impact is in the correct direction, which is a reduction in the mercury levels in the Arctic Ocean. You will note that sea ice and primary production are related. When we were doing this work, we did not have the model in place to have them coupled. So we're looking at it independently, but we know that as the sea ice is changing, that it also impacting the net primary production. So that's actually something that my students are working on right now, is developing like coupled framework that actually will combine all these different changes, and also looking at what this means for methylmercury production. So we had a summer intern, Maya, who is a computational biologist at UCLA this summer, working with my PhD student, Theodore Vincent, and who is modeling sea ice and particle aggregation to really help us better understand what's happening. And that's some of their preliminary work where they're looking at changes in sea ice over time and how it impacts energetic mercury export in the Arctic Ocean. As you can see, we are seeing more export and removal of mercury from the surface ocean as we see that decline in sea ice. And I guess it's kind of a positive spin on this. I don't know. But on this, I will say thank you for your attention, and I will take some questions.
Perfect. Yeah, thanks, Amina. That was great. I guess maybe going back to your work in Labrador, one of the things you touched on was how difficult it is kind of to do mercury research in the context of food supply security in more remote communities. Do you have any kind of tips or things that you use on how to actually communicate those results to the communities in terms of balancing the risk versus making sure that people continue to eat their traditional food?
Yeah, so one thing that I did not know at the time, I know better now, is but we kind of did that. Well, what I did do is having somebody who is on the ground, who lives with the community, and just explain what our founding are, and have somebody who is connected with the community to actually transfer the information in a balanced way through how they understand what we are saying. I think it's really, so our issue was, and still is that overall, I think the traditional foods are better than alternatives. That, you know, if people could switch to, you know, whatever, you know, healthy alternatives, then that would be okay. But that's not happening for many socioeconomic reasons, right? Really expensive to have access to, you know, fresh vegetables. Sometimes they're not even there. So having, you know, food from the land is still the best option. So I don't think that we can just tell people don't eat it, which is what some of the people would try to suggest at the government level, where they're like, oh, just issue food advisories and people stop eating. It's like, okay, but what are you going to eat instead, right? You can't do that. So I think it's just being very clear from the get-go is continue to eat this. This is not the effort shouldn't be on what people should or should not eat. The effort should be, can we make sure we don't increase the mercury level? Like, that's what we want to see. Can the industry or whoever is doing something there not do what they're doing or modify what they're doing, so we can make sure that the mercury levels does not increase in the system? I don't know if that's clear.
I think we have a follow-up question from Colette Wapnitz.
Hi, Amina. Yeah, thank you very much for a super interesting talk and a very clear presentation. I had a follow-up to Kira's question because it's something that I grapple with a lot working with communities and finding these sort of, as a scientist, really interesting things or results in terms of, you know, concentrations of pollutants in fish, but then also realizing the importance that these fish and invertebrates and other animals have in people's diets and livelihoods activities as well. And so I was wondering whether you're engaging at any point as a scientist or as a communicator with some of these communities, pointing out exactly what you say, because I think a lot of the time the message is very clear, like, oh, you have methylmercury, it's dangerous, don't eat it. But actually, in a lot of cases, not eating it is way, way worse because of the DHA and EPA fatty acids composition of these fish to their health. So do you engage at all with some of the policy dimensions of this and mitigating some of these sort of drastic reactions, or are you more taking a sort of step back from that?
Yeah, no, I'm not engaging in that. And in a way, it's not by choice. I think it's mostly through the circumstances of, you know, the academic requirements, right? So it's something that I definitely want to engage more with. So actually, I've had some colleagues where we sat down and thinking about how can we write this down? How can we, you know, can we transfer this information, right? And I've one of the reasons I went to NSF after my postdoc was that I wanted to connect with the federal government people who were engaged in this type of conversations, who were engaged with the communities, and think learning about how exactly do you pass along that kind of information without making a mess, right? So that's something that I'm really concerned about. And so in a way, I stayed out of it out of fear that I'm not equipped enough to say the right things the right way. And so I want to do this. Now the question is how to learn to do it well. And this is something that I'm actually really interested in on a scientific aspect, is how do we as geoscientists learn to walk this line, like not get excited by our results, and be able to kind of like broaden our own understanding of how our results fit in people's lives. And sometimes we're like so narrowly focused that we kind of lose the big picture and forget that there are other things that people have to balance. So long answer to your question is no, because I haven't yet figured out how to communicate this well.
Thank you. I think the next question is from Andrea Frommel. Can you ask the question, Andrea?
Yeah, thanks so much for a great presentation. I mean, I really enjoyed it. I had a question. You had temperature in your models, but is methylmercury also pH dependent? And do you incorporate that into your modeling of emissions and the way it reacts in tissues, carries up the food chain, etc.?
Yeah, so yes and no. So the pH question is interesting because I don't think it comes up in terms of, at least to my knowledge, I don't think that it comes up in terms of methylmercury production in the oceans, because we are kind of still in that narrow band of pHs in seawater that is not, I think, drastic enough to make a difference in terms of production. Although you may see it if it impacts the microbial communities. And I don't think that at this point we have the capabilities to measure methylmercury in such a good and repetitive way in the ocean that will capture the differences driven by a change by a 0.01 pH, right? So I don't know if you see what I'm saying there. However, where I know that Yang Xu has been working on, who is one of my collaborators, is looking at how changes in pH impacts phytoplankton community structure. And so if you have a change in phytoplankton community structure and methylmercury uptake is really strongly driven by the phytoplankton, because that's the first step of accumulation. You go from about 10, the concentration to a 10 to the 4, 10 to the 5 increase in the phytoplankton. So if you're changing your phytoplankton community and the size of that phytoplankton, you may impact methylmercury moving up, but also export, because if you have less calcifying organisms, then you will have less export, and things that don't have a ballast is not going to be exported as quickly. So I think there is some sensitivity there, but not something that I think at this point is going to make a huge difference, at least in the short term. Like longer term, maybe in 100 years or something like that, maybe.
The next question is from Anfal.
Thank you for your presentation. It was very informative. I have a question. Did you face any complications collecting data? You mentioned you took some surveys and the fishing numbers from the fishers or fishermen, and you also touched up on taking hair samples, and did that help? Can you briefly just talk about that?
Yes, I can. I think that one thing that made a huge difference, and I kind of alluded to that early on, is that this was a United government-led study. So they wanted to do this. They were really interested in getting the data. So they were really doing all the legwork in terms of making sure that there is like large participation from their people, the communities that they're like. So it was really driven by them. So it made it easy for us because we didn't have to do all the legwork in making sure that we are communicating why we're doing what we're doing. That was all done by the Nazi government and their, research. It wasn't like research, but their environmental health, environmental science staff, right? So I don't think this is something that I would be able to do by just going and knocking on somebody's door and be like, can I take your hair sample?
Thank you for this answer. I had a small question. When you were analyzing the IPCC models and the scenario, did you analyze different type of emission scenario on the impact of mercury, and what was the result, the different outcome of the different scenario per CMIP model?
Yeah, so are you talking specifically to that emissions?
Yeah, the RCP
data?
scenario.
Yeah, I did not do that. So that was from Noelle's group.
Okay.
So, so that was Noelle and Helen Ango that did the analysis for the different emission scenario with the models. And then I was on the oceans modeling team. So they provided us with the data. So I'm not sure how they do that.
I know this is also coming from another student who just was going to save it for later, but I think it's important to address the big group. Did you work with community to co-develop an agreement or understanding for the storage and destruction of the biological samples, given that Indigenous communities have traditionally had misused biological samples that have led to numerous human rights cases? Was that something that you were thinking of?
Yes, I'm thinking about it. So the reason, okay, backing up at this specific project, so that part, so the collection of hair samples as well as the analysis that was done by my collaborators, Ryan Calder and I think Maling Lee was involved in that. So I'm not familiar. So there was an IRB process that you have to go through Harvard to make sure that all of these aspects are covered. I'm not sure as to the specific details of what they agreed with the national government in terms of sample preservation, but I'm happy to find out, and I'm sure that they followed protocols. I am aware of this and sensitive to that. So one of the things that I did when I was in the Office of Polar Programs is writing the principles for ethical research in the Arctic. And that was like one of the main points of that is, who owns the data preservation, make sure that this remains within the communities, that they have full control of their biological samples. And the reasons for that are the ones that you mentioned, which were there's like a long history of misuse and abuse of samples that have been provided. And that's also one of the reasons why I'm very hesitant, and sometimes like I've been really thinking about this for a long time, and I'm only interested in a one-way thing. Like I'm happy if people come to me and say, we want to do this research, this is what we're interested in. I'm less concerned, I'm less interested in reaching out to people and say, I want to do the research because of these interests, because I think that it should be coming and be driven by the communities themselves and what their interests are. So yes, this is a really important issue and that I'm aware of that.
Thank you.
Perfect. If there's no other questions, I guess we'll end it here. And thank you so much, Amina, on behalf of the IOF. I think you touched on a lot of themes that a lot of us think about and kind of brought them all together. So that was really great.